Okay, today's lecture is going to be on diagnostic clinical microbiology. This is an important field for the allied health. Okay, a couple different things we're going to look at today is how to collect specimens. So what's the appropriate methodology for collecting specimen, depending on where that specimen is. Then we're going to look at processing these specimen. And we're going to look at different types of assays. So we have point of care detection assays, or where we're sending out our samples to a laboratory for more sensitive and specific testing. We'll also look at different types of algorithms and run algorithms ourselves to be able to distinguish a microbial pathogen. Let's go ahead and look at sterile body sites. So the ones listed here are sterile body sites, meaning that if we sample from these areas, we would not find microorganisms. So there's not a lot of them, but there is our blood, cerebrospinal fluid. So it's the fluid around our brain and spine. Pleural fluid, synovial fluid, that's like in joints, in the synovial pocket, peritoneal within the body, and any kind of tissue found inside an organ. So these are the areas that we consider sterile. Anything associated with the outside of the body, or if we're looking at the gastrointestinal tract, would be colonized with microorganisms. But mostly sterile body sites are listed here. The microorganisms that we find within the body can be aerobic or anaerobic, meaning they can be dependent or not dependent on oxygen, and oxygen may be relatively dangerous to them as the reactive oxygen species can accumulate. Looking at how we can sample here, we see this is a way to do sample collection for sterile body sites. So here we see a tube where we go out, we take the sample and we put it directly back into the tube, which is going to be which can be an anaerobic tube for growing microorganisms that can't be exposed to air. Moving on, we have different types of culturing mechanisms and different types of sampling mechanisms depending on the tissue type of interest. If we're looking at culturing microorganisms from the blood, we would do a venipuncture, so puncturing through the blood. We can do a blood culture from this. A couple different ways to look at blood would be directly underneath the microscope, which is what we see in the bottom right. We see blood taken to a light microscope. You can see small microorganisms next to our red blood cells there. We can also grow the microorganisms with a fluorescent marker to look for specific pathogens. If we're looking at cerebrospinal fluid, CSF, this would be a puncture, so a lumbar puncture, so a puncture to the back in which we extract with a needle, in which we can extract the serum and then culture it, or we can look at it directly with microscopy. Looking at pleural synovial or peritoneal fluid, this would be needle aspirations. The needle will be placed in, extract the sample. And again, we can do microscopy or culturing to look at these samples. Looking at sampling from non-sterile areas of the body, this is what we're probably more familiar with. This is a nasopharynx sample or swab. Nasopharyngeal swabs go quite far back. This is something you may have experienced during the COVID pandemic for sampling. This was not just an intranasal swab. A nasopharyngeal swab goes quite far back to gather sample. We can also sample our skin or other abscesses in the body. So if we have a cut and we want to see if the cut does have an infection growing, we could even use needle aspiration deep into a wound to check if we have microbial growth and infection occurring. The lungs is also another place of interest to look for microorganisms. So we have what we call sputum, which can be coughed out of the lungs. We see this in the picture at the top right. That is content from the lungs, and we can assess it for microorganisms. So we can look for things like tuberculosis or pneumonia within this sample coughed up from the lungs. If patients are unable to produce sputum correctly, we can do a bronchoalveolar lavage or BAL, BAL. which is where we actually put solution down and extract it again and see if we can find microorganisms, bacterial microorganisms within the cultured serum. Okay. Looking at urine collection, again, this is a non-sterile site. Urine is in a anaerobic environment, so it would be growing without oxygen in a closed environment within the bladder. There's a couple different mechanisms to looking at anaerobic microorganisms collected from the bladder. So one, we can look at doing a clean catch mechanism. So a clean catch is where urine flows through the urethra and cleans out some of the microorganisms that are typically found within the urethra or on the outside of the skin. And then the sample is collected partway through to reduce the amount of our own microbiota getting into the sample. We also have the use of a catheter to gather a clean sample. Here we can see a catheter being used, catheterization, all the way up to the bladder so that we can take a clean sample from the bladder. Okay. When we handle biological specimen, there's different levels of PPE that need to be assessed. We have our biosafety levels here, 1 through 4, 1 being the least dangerous, 4 being the most dangerous. BSL level 1 and 2 is what we see most teaching labs to be, especially if they have Staphylococcus aureus in them. They fall into biosafety level 2. Looking at safety practices and safety equipment, we see that these are increasing from BSL 1 to BSL 4. So here we see open bench work with BLC 1, which is done in a common teaching laboratory. Some microorganisms we may need to have a biosafety cabinet for any kind of potential aerosols that we're breathing in and more protective clothing. Moving on to biosafety level 3, specialized clothing, controlled access and directional airflow so that the air is not leaving the lab. So we want that pressure inside. And we also have biosafety cabinets here, which is typically where work is done. On a biosafety level 4, here we're working with dangerous pathogens. These normally have airlocks, showers, and special waste disposals. And the safety equipment that we would see would be the highest level of safety equipment. So this is when we see people in those large suits. They actually have positive pressure, meaning that if there was a hole in the suit, it would prevent microorganisms from coming into the suit. The positive pressure would push outwards, preventing microorganisms from getting in and contaminating the person. We also have double-ended autoclaves and filtered air within a BSL level 4. Let's go ahead and talk about how we can assess microorganisms. So we can culture them, we can look at them underneath a microscope, and we can do biochemical testing to identify them. With all of this information, we can build essentially an algorithm to figure out what our organism is. So biochemical tests are typically done in the laboratory for unknown organisms. This is kind of like simulating an environment of somebody coming into the hospital, grabbing a sample, running biochemical tests from plating on media like differential and selective media, to culturing the microorganisms, and then to assessing with algorithms. Here we can see on this picture down here a fastidious microorganism. This is a microorganism that doesn't grow well, so they had to use specialized agar. So this is chocolate agar. Chocolate agar actually has lysed blood cells, so it's adding additional nutrients for microorganisms to grow. Okay. Looking at the algorithms to identify them, we have to gather the data first. What we see in this image here is the typical assessment test, the biochemical tests that can be done in lab to identify a microorganism. Here we can look at identifying a gram-negative enteric microorganism, one from the gastrointestinal tract. Here we see different tests like indol, VP, urea, hydrogen sulfide, citrate, glucose, mannitol, sorbitol, arabinose, gelatin. So we have all these different types of tests on these test strips. We see test strip A, B, and C. What we see with A, B, and C is that all of this is a little bit of a media, biochemical media that's essentially inoculated as we move through, that these look different. So ONPG, going from B to C, we can see looks completely different. So as we move through here, we can see different kinds of chemical reactions. We can see a positive reaction here versus a negative reaction here for mannitol. So that would mean this microorganism breaks down mannitol sugar, while this one does not break down mannitol sugar. So by accumulating all of this data, like this one's urease negative, this one's urease positive, we can identify this specific gram-negative enteric microorganism. Looking at something like this is quite different than what's done in a teaching laboratory in the fact that everything is all on one strip. In a teaching laboratory, these are all normally different plates, different broths, different slants that are used to inoculate. Okay, looking at gram-negative microorganisms, we have non-enteric ones as well. So ones that are not associated with the gastrointestinal tract. And we would have a different set of biochemical tests that we would use to identify these specific microorganisms. Okay, let's go ahead and look at dichotomous keys. This is essentially a problem-solving algorithm, and it goes dichotomous because we're asking basically a question with two answers. So we're starting out knowing that we have a gram-negative rod. So this could be identified from a gram stain. That's typically your first step in an unknown. So gram stain, you see the gram negative for this one. So it's going to be a pink color. And then we know that it's a rod-shaped microorganism from microscopy and understanding morphology. Then we go to do a glucose test. Following the glucose test, if it's negative, well, we've already figured out what our species is. It's Pseudomonas, a gram-negative rod. If it's positive, on the other hand, we need to do additional testing. This is where ONPG, a lactose test, could come in. Let's say this test goes ahead and ends up negative. Well, then we have further testing to do, like a hydrogen sulfide test. Let's say that one's negative. Then we could do a citrate test, and let's say that one's negative. Then a sorbitol test, and let's say that one's positive. All of a sudden, we know it. We have Yersinia pestis, the black plague microorganism. So going through these based on what the results are, you build basically this chart, this tree that you can follow down and figure out what the microorganism is. So with this ONPG, if it ended up being positive, we could do something like an indol test. Let's say that was positive. We do a VP test. Maybe that's negative. We would do a citrate test. Let's say citrate was negative. We could do a urease test. Let's say that was negative. Then we find out that our sample is E. coli. So we can see all of the microorganisms down here, and how we get to them is based off of this dichotomous key. And that's how we assess unknowns in the laboratory, is using these problem-solving algorithms from biochemical tests. Okay, this can be automated in a laboratory. So we talked about teaching labs, we talked about inoculation of media. We also see here the automation of this process on a 96-well plate. So if you were to zoom into this picture, you could see things like VP here. It's a little bit hard to read. VP, ERA, sorbitol, urea. So we have all of these biochemical tests on this plate. So they're grouped into these patterns, and they can be read through processing. We could sample them this way, and we could get all of the data at once. And this being machine read means that there's less human error in identifying a microorganism. Working in groups with other students, you might think a sample is positive and another student might think it's negative. This way we get around the problem with human error. Okay, let's go ahead and look at the same thing with a gram-positive microorganism. This is pyogenetic cocci. So we've already done a gram stain and we see it here. So the gram stain was positive. It's taken from a lesion. We have blood agar, CNA, and McConkey agar. It was plated on different types of media. And then we see we can do a gram stain. Looking at something like McConkey agar is selective and differential, and it's selective for gram negative. So the fact that we see no growth shows that we're gram positive. We also have a gram stain showing us that our sample is gram positive. It shows that crystal violet purple. So we have secondary verification that we have a gram positive organism. Gram stain is one of the most critical staining procedures that you do, and it's initially done with an unknown, and it really sets off the algorithm whether we're going gram negative or gram positive. So it's important to get the gram stain right. It may need to be done multiple times to assess, and backup tests like using McConkey agar to verify that this is a gram, positive organism in this case is important. Okay, So let's go ahead. Here's our sample. This is a microscopy image. Then a catalase test is done. And this dichotom is key. We can go negative or we can go positive here. So in this case, we see that we went negative. So we don't have the bubbling from a catalase test. Then we can go ahead and do hemolysis on blood agar. Is it alpha, beta, or none? Here we see that we have beta hemolysis. So we see those red blood cells are lysed open. We do bactracin and we see if it's positive or negative. We see bactracin was positive. The microorganism cannot grow around this impregnated paper that has the antibiotic bactracin on it. That lets us know that this is Streptococcus pyogenes being sensitive to bactracin. And that's how we identify our organisms. So if you were given these terms, you'd be able to follow it down. So for example, if I said it's catalase positive, coagulase positive, that would leave us with Staphylococcus aureus. We want to be able to work our way through these charts and be able to build these charts. Other microorganisms that are a little bit more difficult to identify could be acid-fast bacteria. These have mycolic acid in them that prevent a gram stain from effectively identifying a microorganism as gram-negative or gram-positive. That waxy, which we see this waxiness here, this waxy layer prevents, again, that stain from being incorporated into the cell. So we'd have to have different biochemical tests, an acid-fast test for things like Mycobacterium tuberculosis or Mycobacterium other species. Other ways to identify microorganisms are more off of molecular means. So here we have mass spectrometry, MS, specifically with Meldotov MS, which is matrix-associated laser desorption, ionization, time of flight, mass spectrometry, Meldotov MS. The way that this works is by adding our cultured sample into a plate. So this is one of those kind of 96 well plates again. Once we add it into a plate, it's actually ionized by a laser off of the plate, and that's where the time of flight comes in. So once it's ionized, all of these proteins move away from the sample at different times. And what we can see here is a mass charge ratio of these proteins. So this is E. coli that we're looking at from a sample. And we're looking at the time of flight results for these individual proteins within E. coli. So this is like a fingerprint of E. coli, meaning that only E. coli gives you this picture. So this is the mass charge ratio from Meldotov MS. So here we see mass and charge ratio. Each one of these is the proteins and their time of flight that we're looking at, the mass charge ratio, and percent intensity. Looking at all different microorganisms, their protein composition is different. So this image that we see here for E. coli is unique to E. coli. If we're looking at a different microorganism, they're going to have a different fingerprint, a different profile from Meldotov MS. It's a great way to identify microorganisms. We also have PCR-based reactions. That means polymerase chain reaction. Again, PCR is a process of amplifying DNA, of gathering more DNA. But we can also do this by individually identifying unique genes by using primers. So instead of cell division where we replicate all of the DNA, PCR targets specific areas of the DNA, specific genes of interest. So here, let's say we want to identify virulence factors from a microorganism. We can look at these different types like type F, type A, type B, type F, and identify them. So we would amplify them with primers that amplify these specific areas, then run them on a gel. And this is gel electrophoresis. What we see here is that the samples would be placed at the top. These samples are negatively charged. DNA's phosphate backbone is negatively charged, and they will move towards the positively charged end. As DNA moves through this gel, this matrix, it's going to be moving faster if it's a small piece. And if it's a large piece, it's going to take a lot longer to move through. So we can basically amplify our DNA. And then we could even use restriction enzymes to cut them up and look at the size of these proteins to see if we have the same ones. Here, a good way to think about it is the small ones move through the matrix because they can snake through. The larger ones get stuck within the matrix. So the larger ones are found at the top. These are the heavy ones. So we see they have more base pairs, like 1,030 base pairs, where these have maybe like 100 75 base pairs. So looking at these pathogens, these markers here, we have type A, type B, type E, and type F. Let's say this is a sample and we see that here it lines up with type A. Because they're the same weight, we can know that this has type A. We know that it has type F as it lines up here, and it has type B, but it does not have E. If it did have E, that DNA would have ran down to that same level that we see here. Let's look at sample number 7. So our sample was placed in here. The DNA moves down. The smaller pieces are going to move down faster. The heavier ones stay at the top. Now let's check for A. We don't have A. We do have a band here, which corresponds with F. And we do have a band here that corresponds with E and a band here that corresponds with B. So we see F, E, and B, which is also here. B, E, and F are corresponding here. Looking at 8, why don't you go ahead and take a look at it and list the types that we find there. So you can pause and think about this one. Okay, and to kickstart this again, we can see this band here corresponds to A, this band corresponds to F, this one corresponds to E, this one corresponds to B. So we have all of these types included here. So PCR can also identify genes that have been horizontally transferred from one species of a microorganism to another. So maybe they're picking up virulence factors that enable them to be resistant to antibiotics. So maybe the genes they picked up confer antibiotic resistance. We can check this way as well to see if there's genetic changes. We also have something called quantitative reverse transcription, PCR. So it's a polymerase chain reaction, again, based off of the same idea. But we can identify even viral pathogens this way. Let's say our viral pathogen comes in with maybe mRNA, or we're looking for a specific mRNA sequence. We can use that enzyme, reverse transcriptase, which is the enzyme that, again, viruses have to go from RNA to DNA. The reversing transcription, as DNA typically goes from as transcription goes from DNA to RNA, it goes backwards in reverse of the central dogma. So we can use that same enzyme, reverse transcriptase here. We can take our mRNA, use reverse transcriptase to build the corresponding cDNA, and then we can go ahead and remove that mRNA and go ahead and build again. We can use an enzyme to make more DNA. So now we can end up with our double-stranded cDNA. In this whole process, if we want to highlight a specific area, we can use fluorescent dyes and quenchers. And this is actually something that I used to do. I used to work at Roche Molecular System in biotech. I went into biotech following my undergraduate degree. Following my master's degree, I went back into biotech, and then I went back into school for my PhD. So here we can see fluorescent dye and quenching dye. This is the same thing, a PCR reaction. We have our primer. Primer anneals to the target DNA, but what we have downstream of the reaction is our fluorescent dye and quencher. This quencher prevents the fluorescent dye from leaking signal, from being observed at the moment. So it's suppressing signal. But as soon as we have DNA replication here, we have our Taq polymerase moving along, it smashes into the fluorescent dye, breaks off the fluorescent dye from the quencher. Now the quencher is no longer suppressing the signal, and we see this bright fluorescence. So every time a gene of interest is amplified through PCR like this, quantitative PCR, we see a flash of light, and an instrument picks up this light. We don't visualize it ourselves, and the instrument tracks how many times we see it. Every time we amplify this and that quencher and fluorescent dye is on there, and it hits that, it breaks off. So we can see the signal slowly increasing over time. And that's what we're going to see here is these signals increasing. This is that fluorescent light. We have patient 1 and patient 2. These are the cycles of PCR. So again, we heat up, we denature the DNA, we add our primers, we drop the temperature, add our primers, they anneal to the specific gene of interest. Then we go ahead and have our enzyme. We heat it back up, our enzyme adds on and amplifies the DNA. As it amplifies the DNA, it hits that fluorescent marker and quencher, breaks them apart, releases light. and continues replicating. So now instead of one double helix, we now have two double helix, two double-stranded pairs of DNA. And then we repeat again with PCR. Well, that's the next cycle. So cycle number 2 be again denaturing. Then we drop the temperature. The primers come in, increase the temperature. The enzyme, the Taq pol comes in, replicates the DNA, hits that quencher and fluorescent marker, and fluoresces again. So we hit a threshold here with patient 1 and patient 2. Patient 1 crosses that threshold sooner than patient 2, meaning they had a stronger signal. So to start with, they had more of maybe this virus or more of this biomarker. So as we see this increase, this logarithmic expansion of fluorescence, we see patient number 1 had a higher concentration to start with than patient number 2. Here we can see cycle number, amount of DNA. We start out with 1 double-stranded DNA. After 1 cycle, we go to 2. After 2 cycles, we go to 4. After 4 cycles, we go to 8, then 16, 32, and so on. And here you can see we go from single digits to hundreds, to thousands, to tens of thousands, to 100,000, to millions, and then we get all the way down to trillions of copies of DNA. So at 34 cycles, we've really made quite a lot of DNA from 1 strand all the way down to trillions of strands. Remember, this is also the same technology for crime scene investigation. The little bit of blood spatter or semen is found at the scene, that DNA can be extracted, just a small amount, and amplified. So if we have 1 copy to trillions of copies. This is how crime scene investigation has gone back through cold cases, picked up that old DNA that was found, and have been able to amplify it and identify maybe unknown victims, these John Does, or maybe perpetrators as well. Or they've been able to get people out of jail by going back and looking at who was put in jail. Does this blood spatter actually match up with the person who's in jail? Okay, let's go ahead and look at genetic fingerprinting. So genetic fingerprinting has to do with microorganisms themselves, whether that be viral, whether it be bacterial. We have things that we can identify on them. So we can take a specimen for PCR and we can go ahead and work with that specimen to amplify it. So let's say a COVID test, for example. We have an at-home COVID test versus we have a PCR COVID test, a qPCR test. The at-home one can pick up COVID, but you have to have a significant amount of the virus within you to pick it up. It can't just be day 1 exposure. On the other hand, PCR is so sensitive, it can pick up just a little bit of the viral infection. Okay, let's go ahead and look at RFLP. This is restriction fragment length polymorphism. So similar to what we saw here with gel electrophoresis, we talked about amplifying specific genes through PCR. We can target that gene of interest, gene A, gene B, gene E, for these types and amplify them, or different variants of those genes. Here with restriction fragment length polymorphism, we can just take the DNA of interest. We can cut it up with restriction enzymes. Remember, they cut at those palindromic sites. Once they're cut up, we can put them onto a gel, run them, and we can look for matching patterns. So here we see these 2 lines are replicates, these 2 are replicates, these 2 are replicates, these 2 are replicates. And we can identify specific portions of the genome based off of gel electrophoresis, which still uses that same method of negative DNA strands run towards the positive end of the gel. These are the wells up here. DNA is negatively charged, so negative and positives attract. The smallest fragments will be able to weave their way through. The larger fragments will get stuck at the top. That's kind of like elephants versus mice running in a forest. The elephants will get stuck within the forest. The mice will work their way through and get through the forest faster, the smaller ones. So the big elephant DNAs, the large ones are up here. Small mice ones are down here. Okay, let's talk about diagnostics. When I worked at Roche Molecular Systems and another company, Introgen, for assay development, assay is just another word for a test, what we really focused on was sensitivity and specificity. And this is really important. This is essentially how good the test is that you're running. How much of the sample can you pick up? So let's go ahead and look at sensitivity. You could ask yourself, if a patient has a disease, how often will this test be positive, right? So let's say you take a sample of 100 people and they have the disease. Maybe some people are on day 1, maybe some people are on day 10, maybe some people are on day 15. Well, with all of these people that do actually have an active infection, how sensitive is this test? How often is this test going to pick up? Is it going to pick up 100% everybody? Is it going to only pick up 50% of the people? The people who just got infected, maybe the viral load or bacterial load is so low within the body as it hasn't started replicating that the sensitivity is not on par. Maybe it's going to skip all those people. So the higher sensitivity we have in a test, the more it can pick up of the people who have the disease, the better it is. Let's look at specificity on the other hand. This is another key player here. What you could ask yourself is, if a patient does not have a disease, how often will the test be negative? Because we don't want false positives. We don't want false negatives with sensitivity, and we don't want false positives with specificity. So that's really important for assay development and design. And this is really what we work hard on in the lab. So for specificity, we want to make sure that our assay is not picking up similar microorganisms. When COVID first came about and people were doing testing with COVID, we actually had some of our tests picking up SARS, people who had prior exposure to SARS, because the viral structure was so similar to one another. So we had people misidentifying, getting false positives because the specificity was not great. We hadn't come across COVID yet. So we didn't know that the COVID tests we were making were also picking up people who had previously had SARS and giving them a false positive reading. Okay. Looking at sensitivity and specificity, if we compare an at-home COVID test with a qPCR COVID test, the sensitivity and specificity is the biggest difference. The at-home test is great because you can get immediate results. But remember, if you did an at-home test and you found out it was positive, they told you to go ahead and get a qPCR test, and that's because it's more sensitive and specific. The same thing happens with even pregnancy tests. If you do an at-home test, they'll send you into the doctor to do a more sensitive and specific test for confirmation. Let's look at point of care testing. So these are tests that you might buy at the store at home, like a COVID test, or maybe it's even something that you go to a drugstore like a CVS, where they have a nurse who's going to go ahead and administer these tests for you that are just quick tests where you get the results back right away. So the problem with the point of care tests, though, is that their sensitivity is not great. Let's say you just got infected with COVID and you take one of these at-home tests, and let's say someone else just got infected as well and did a qPCR test. Well, qPCR test might show a real positive, but the point of care test, on the other hand, might come back with a negative because it's not sensitive enough to pick up that minuscule amount of the virus, where PCR, we saw, can pick it up from maybe like very low numbers if we're looking at this. So we can amplify so easily. So again, the more you come in with, the faster you're going to hit those thresholds and move up, the more accurate those tests are. But point of care is also great because we can get results right away. So we do sacrifice sensitivity a little bit. Let's go ahead and look at how these tests work. We have two major lines on them, control line and test line. We have to verify that the control line is working. Then the test line is whether you're positive or negative for the specific organism you're testing for, or pregnancy test, the hormones that you're looking for. In these type tests, you typically drop a specimen here. So for a lot of the COVID tests, this was a nasal swab that you put into a buffer, and then you added that liquid into the sample. And what happens is the liquid actually moves through the test strip. So here is our drop of specimen. It's going to contain, oh, here we're looking at pneumonia. So we have pneumonia, the microorganism here. So portions of the microorganism are found in the sample, but we also have other components that are just part of that nasal swab, or we'll get lung swab, or whatever you've done here, oral swab that you're testing, right? So in this test here, we hit a couple different things. We see our anti-pneumococcal gold-labeled polyclonal antibodies. So these antibodies are going to interact with these markers that are coming through from the sample. So let's say it picks them up. Here we can see picking them up. And then on the test line, we're going to have these anti-pneumococcal rabbit polyclonal antibodies. So these are actually stuck on this test line, this portion. And so when these come along with a pneumonia sample, they're going to go ahead and bond. If they don't pick up a pneumonia sample, they will not bond. So the antigen is not there. We'll not see bonding on the test strip. This is what gives us the positive or negative results for, let's say, the microorganism or virus itself. On the other hand, the control line, the sample continues moving past the test line. And our control line is what actually captures the antibodies that don't have anything bound to them. So this is our solid phase goat anti-rabbit IgG antibody. So we see we have the anti-pneumococcal gold-labeled rabbit polyclonal antibodies bonding to the solid phase goat antibody IgA antibodies. So here they're going to bond. So this is what allows you to have a control line that comes up positive. The positive control doesn't come up. It means this assay didn't work, that these antibodies did not move all the way to the control line, meaning it's not a valid test, and we can't depend anything on that test line. Control line has to be there. So again, we do lack sensitivity in these tests, but they are a good way to first look into this. Another thing to look into, a lot of these at-home tests, when we're reading through these, students ask things like, why are these rabbit polyclonal antibodies or anti-rabbit or goat anti-rabbit? These antibodies are actually generated in animal models, so in animals themselves. If we want an antibody that works against pneumonia, well, we have to inject them with pneumonia. Then their immune system generates antibodies. The animals are bled, and these antibodies are extracted from them. So it's not necessarily animal testing, but it's manufacturing of proteins like antibodies within living systems. So we're taking advantage of the immune system of the animal. So if an animal, let's say the goat, is making these anti-rabbit antibodies, we've actually injected the antibodies of the rabbit into the goat and had them generate antibodies for us. So this is the most common way that these tests are made, is by these polyclonal antibodies are made in animals themselves, or just clonal antibodies made in animals. And we have other means of making them today, but that's why we still see these terms, rabbit or goat, on here, because they're generated in these animal models that are then sacrificed following the extraction of the antibodies from their blood. Okay, so again, these at-home tests work great for quick turnaround, but they lack sensitivity. So let's look at advances in point-of-care tests. Some of the benefits of this is maybe you just go into a CVS or you buy a test from a drugstore, take it home. There's no culturing of these microorganisms involved. You're actually using the sample itself and testing directly from that. Following that, let's say you come back positive on something, antibiotics can be prescribed right away. So point of care, you're going maybe into a CVS, seeing a nurse, they're doing the test. Well, you can walk out of there with antibiotics that day. You can also avoid taking antibiotics. Let's say it comes back negative. Well, then you can avoid unnecessarily taking antibiotics as well. So here we can see also quick chain of infection. If we're looking at the spread of disease in maybe a nursing home facility or in the hospital, we can do these quick tests and identify very quickly where this disease has gone. Is it in the doctors? Is it in the patients? How is it getting room to room? So it's a very quick assessment that can be done. And we can notify patients right away. Typically, if a doctor goes into the doctor's office and if a person goes into the doctor's office and takes an assay and then has to find out weeks later or a week later or a couple days later whether they have an infection or whether they don't, it's hard to get in contact with those people again. So it's hard enough to get people into the doctor's office, then hard enough to get them to come back in order to receive the proper treatment. So with point of care, treatment can be administered right away. We don't have to wait for people to come back. Some disadvantage of point of care treatments, though, is that we don't understand if the microorganism that was picked up is even sensitive to the antibiotics that are being prescribed. So we're just really going off our understanding that this specific organism is typically sensitive to this antibiotic. We know through horizontal gene transfer that microorganisms can become resistant to specific antibiotics. So we could end up with a resistant strain and it not be caught right away. If the person starts taking antibiotics that are not effective, the pathogenicity life cycle of the microorganism will continue for longer, and the infection will continue to get worse and worse before we get treatment for the person. Looking at point of care tests, another problem is that the clinician who's constantly doing this is in contact with people who are sick. So there's a risk of infection from them as well. So it's a high risk job. Another problem is that let's say you take a point of care test, it's positive, you get an antibiotic, you go home, you think you're solving the issue. The other thing is that there can be multiple infections, not just one. So you think you're addressing the issue, but by not doing a more comprehensive, holistic analysis of what's actually going on, we may miss that there could be 2 different infections, 3 different infections going on. Okay, let's get to some of these questions here. Here is our algorithms. These algorithms are fun to go through, so you can test yourself on these. This one says, what is the identity of a gram-positive cocci that is positive for catalase and positive for coagulase? So go ahead and pause the video here and figure out which Streptococcus species this is. Okay, we're going to go ahead and look at the answer now. This is Streptococcus aureus. Let's go ahead and see how we figured that out. So here's a lesion. They've sampled it. They've ran it on blood agar, CNA, McConkey agar, and it is negative. We know this is gram-positive, so that's typical for gram-positive not to grow on the selective media McConkey, which favors gram-negative organisms. Following a gram stain, we're visualing the image from microscopy where we see morphology and gram. It's gram-positive cocci. You can see this from zooming in. Okay, now it says it's positive for catalase and positive for coagulase. So let's see here. The catalase test, it said it's positive for, so we move in this direction, right? Positive, we see the bubbling here. Then it says for coagulase, it's also positive. So we go here, and that leads us to Staphylococcus aureus, right? If it was negative, we could do novobion testing and see if we're resistant or sensitive to the antibiotics. But here we just stop at Staphylococcus aureus. Okay, question number 2. For which of the following clinical samples would it be necessary to perform an initial culture using a selective culture medium to look for the presence of a pathogen? Go ahead and pause here, think about the question. And the answer to question number 2 would be a stool sample. So the reason why the initial culture, you'd want to do selective culture medium is because a stool sample has a lot of natural microbiome within it. So all of our gastrointestinal tract has our microbiome. Blood, cerebrospinal fluid, and synovial fluid should be considered sterile. If we have a sterile sample, we wouldn't need really selective culture because we have so few microorganisms anyways that even just the presence of a microorganism is a concern. Stool, on the other hand, is littered with microorganisms from our own gastrointestinal tract, many of them healthy, but maybe we're looking for Clostridium difficile or something else. We could use a selective culture that maybe selects for gram-negative enteric microorganisms and not gram-positive microorganisms. Okay. Question number 3, another algorithm or dichotomous key. Which of the following biochemical characteristics is associated with Yersinia pestis? Go ahead and pause and think about it. Follow the chart. The answer to question number 3 is ferment sorbitol. So we're looking at which of the following biochemical traits is associated with Yersinia pestis. So we can actually back calculate here, right? We go to the microorganism itself and we work our way up. We know it's positive for sorbitol, but we don't see that listed. We know it's negative for citrate. So it doesn't utilize citrate. That's the exact opposite that we see here. And then as we move up, we see it's negative for hydrogen sulfide production. Okay, so knowing that it ferments sorbitol is from the very beginning here. Sorbitol positive, ferments sorbitol. So we can back calculate, make our way up. Yersinia pestis is positive for sorbitol, so it ferments sorbitol. Moving up, it is negative for citrate. So again, does not utilize citrate. And then it's negative for hydrogen sulfide production. So here it says produces, it does not produce hydrogen sulfide. So the only thing it does is ferment sorbitol. Here we see produces urease. That's on the other branch of the tree that we're not going down, as we're only focusing on Yersinia pestis and back calculating. Okay, question number 4. A diagnostic assay that gives a high number of false negative test results has a low, go ahead and think about this, looking for a false negative test result. This would be sensitivity. So a false negative means that someone is actually positive. They have the microorganism of interest, but the test comes back as negative. So it's a false negative because they should have been positive, meaning the test is not sensitive enough to pick it up. So maybe the person just got COVID, they took one of those at-home tests, and the sensitivity is low at those at-home tests, those point-of-care-like tests. So it doesn't pick it up and it gives somebody a false negative. Okay. Let's see, question number 5. The Clostridium botulinum isolate in lane 6 produces the blank toxin. Go ahead and pause and think about this one. We're looking at lane 6 here, so our sample's inserted here. And the correct answer is A, B, and F. So we look at these lines. A corresponds here. We have B corresponding here, and F corresponding here for lane 6. We do not have E. This is what it would look like if we did have E as well. So we have A, F, and B. Okay, we've now gotten on to our section objectives, again, to be done at the end of lecture, to review these points with others or with yourself. And that brings us to the end of the lecture.